Welcome, everyone. We're going to go ahead and get started. I appreciate everybody's time and attendance at today's ISV webinar. It is my great pleasure to introduce to you Dr. Emanuele Montomoli from the University of Siena. He lived most of his career at the University of Siena or in the Siena area. He has received his bachelor's and master's degree from the University of Siena in physical and mathematical sciences, and he earned a doctoral degree in biochemistry and enzymology at Siena University. He was then appointed as a faculty member in the Department of Medicine and Surgery. And in 2016, he was promoted to full professor in the Department of Molecular Medicine at the University of Siena. He serves on several scientific boards, including Toscana Life Science Foundation. He also serves on the International Society for Influenza and Other Respiratory Viruses as a scientific board member. He's participated heavily in that society, hosting several junior seminars and training sessions. And his research is primarily focused on epidemiological and microbiological research associated with respiratory viruses. He's authored over 200 scientific publications. And really today's talk is right up his alley of what he has been doing for his career, which is looking at respiratory surveillance on influenza and serology of other respiratory viruses as well. So we welcome you. I appreciate you being able to present today to the Society. I would like to remind all members on here and all Society members that the ISV Congress is scheduled for October 21st through 23rd in Seoul, South Korea. The registration site is open and abstract deadline is on June 17th. So we're hoping that many of you will be able to attend the meeting and attend the Congress and present your data there. So now I turn it over to Dr. Montomoli. We welcome you and you can lead your presentation.
Okay, thank you very much. Thanks a lot, Ted. Can you hear me very well before to start? And you can see my screen as well, I believe.
Yes, we can. Very well.
Okay, very good. Feel free to interrupt me anytime in case that we have some technical issues, but I hope no. So let me thanks before my friend Ted, who invited me to present for this webinar, and let me thanks the ISV as well. So it's a big honor for me to present about my research field on serology for influenza and other respiratory viruses. As Ted said before, I have a position at the University of Siena, but more of all, in 2009, we set up a private company as well, which is called Moderator, where we are doing serology for several preclinical and clinical activities as well. 2 slides only just to say what we are doing now in our company. Of course, we are doing, as I said, which is underlined in red, serological analysis for vaccines licensing, following the EMA guidelines, FDA, PMDA, and ANVISA, and several other regulatory agencies. And we are doing optimization of analytical tests for biological samples as well. And we are doing preclinical analysis and support for animal testing, in addition to the central lab activities. We become quite famous in the last years because we did the SAPI recognition as one of the SAPI reference lab. And in addition with SAPI, we work together with several international organizations as IVI and PATH and the Gates Foundation, and with several companies as well. I have listed here some of those which are not confidential, but there are much more than this. Let me go quickly to my presentation. Today, we are talking and we are discussing about the serology for influenza, and in case if we have time for other respiratory disease vaccines as well, and correlates of protection. That's the first question. So some of you probably expect to discuss and to find a cutoff just to identify a protection for influenza or to identify the right assay to establish the correlates of protection. But before to start, we have to say that correlates of protection is a different thing, because correlates of protection means the immune response that is responsible for and statistically correlates with the protection. And it depends a lot from the assays that you use to evaluate the immune response. But when we say protection, we should define if protection against infection, if protection against disease, for instance. And when we say protection, which population should be protected, if pediatric, adults, elderly. So it becomes very, very complicated. Some years ago, I was one of the coordinators for an European project that originally, which is called FluCop, and originally was set up for to establish a correlates of protection. In the end, we decided to stay only on the assay optimization, harmonization, and validation, because it becomes impossible to establish the right correlates of protection. We should define too much variables probably to define a correlates of protection. But of course, the basis to establish a correlates of protection is to have a good assay to establish the immune response. Again, that's another definition of correlates of protection, which is done by the FDA. Again, FDA define the correlates of protection as a laboratory parameter that has been shown to be associated with protection for clinical disease after adequate and well-controlled clinical trials. Is an immunological correlates of protection is most useful if clear qualitative and quantitative relationship can be determined. And of course, to have a clear qualitative and quantitative determination, we need to have a very good assays. In Europe, the situation for influenza is quite similar, but we have defined in advance already in 19, as you can see, these are the guidelines released in 1997 already, that for influenza, they say the European Agency for Evaluation of Medical Product define that influenza vaccines must be, the immune response to the influenza vaccines must be evaluated using hemagglutinin inhibition and single radial hemolysis. Some years later, we maintain the HI and the single radial hemolysis as official methods, but we added the neutralization and as an official assay for vaccines evaluation. And we suggested to investigate the T-cell response as well, and the role of the neuraminidase as exploratory endpoint. So telling that we have a quite clear panel now of assays that we should discuss just to evaluate the immune responses versus influenza vaccines. So very quickly and very briefly, let me define and let me see what are the advantage and the disadvantage of all the assays that we have cited before, and then we are going to quickly describe in details later. The hemagglutinin inhibition is very well known from almost everybody who works in this field. The hemagglutinin inhibition, we have some slides later on the hemagglutinin inhibition, has the advantage that is suitable for screening a large number of samples. I mean, in our lab, staff of 10, 20 employees can analyze 4,000 clinical samples in duplicate or sometimes in quadruplicate per week as well. So we can do a lot with hemagglutinin inhibition. It detects antibodies that bind around the globular head of hemagglutinin and block the hemagglutination. But we come back later on this point because we have some nice cartoons that can explain what I mean. Has a good correlation with microneutralization for seasonal strains, and only biosafety level 2 labs need for pandemic strain as well, because we will use it, but again, we will show later much more in detail. We use the inactivated viruses as the antigens to do the analysis, and is EMA and FDA approved, and is, let me say, approved almost everywhere in the world. I mean, we have done a lot of clinical trials for the Japanese PMDA agencies, and the HI is usually considered as one of the gold standard for the vaccines evaluation. The single radial hemolysis is a little bit less known, but it's very easy for the people who's confident with the lab procedures. Is the classical complement fixation. But let me discuss later about the advantage of the single radial hemolysis. It has a good correlation with the microneutralization for pandemic strains as well, particularly for H5. And again, we need biosafety level 2 only also for pandemic, because as original antigens, we can use only inactivated viruses, and it's EMA approved. Sometimes it's not very well appreciated in the US, but is appreciated for the pandemic strains, and is widely used in Asia and in Japan also. The neutralization, of course, is one of the gold standard that is used for confirmation. It titers only the functional assays, only the functional antibodies, sorry, and we will discuss later about the differences between functional and not functional. It's not easy, unfortunately, to screen a large number of clinical samples because, I mean, sometimes, particularly, there are, I will describe later that there are two different procedures. One is the ELISA reader and one is the cytopathic effect reader. In case of CPE, of course, we have to read under the microscope, and sometimes it's not easy. I mean, it's easy, but we need a lot of eyes, let me say. And for this reason, it's not suitable for screening a large number of samples if you don't have a qualified personnel in lab. Of course, we have to use live viruses. And in case of pandemic strains, we have to use under the biosafety level 3 containment. And this could be another limitation. And it detects antibodies that bind around the globular head of the virus and block the virus attachment and entry in the cells. We don't have a clear correlates of protection established. Let me say, more than correlates of protection, we don't have a clear cutoff of protection already established. In addition to those methods, we have a huge panel of other assays. Of course, we can use ELISA. I don't spend too much time on the ELISA because everybody knows. We will discuss later about the pseudotype neutralization, which has a lot of advantage, and I will explain how we can set up some good pseudotype for pandemics, particularly for pandemic strains. And we cannot forget about the role of neuraminidase, and of course, we should analyze the immune response versus the neuraminidase. And the new frontiers are the multiparametric assays. Very recently, several companies are looking for to screen several parameters using the multiparametric assays as the MSD or the Luminex or something like that. But we have time to discuss about that. Let me start with the more easy. That's the hemagglutination. Couple of minutes just to explain, but everybody know about the hemagglutination. So let me try to have a pointer in case. Okay, it doesn't work, no problem. So that's the hemagglutination, the hemagglutination inhibition procedures. So as you can see, we have the virus and the red blood cell, the animal's red blood cells. In case that the virus can bind, let me check. Okay. Now you can see my pointer as well. We have the virus and the red blood cells. In case of no antibodies, the virus binds the red blood cells, and we can see an hemagglutination on the microtiter well. In case of antibodies, the antibodies bind the virus, and of course, the red blood cells are not hemagglutinated, and we can see a bottom on the microtiter well. So the cutoff of protection is a dilution equal or greater than 4 as a dilution factor. Let me try to explain better in the next slide. So can you see my pointer or the red pointer? Can you see? I think that you can see, correct? Okay, good. So as you can see here, we have here in the first 2 columns a negative samples because we don't have inhibition of hemagglutination. This means that we don't have antibodies. And what happened? Happened this one, we have hemagglutination. So here the samples is negative. These samples, if we consider 1 to 40 as a cutoff of protection, is high positive in duplicate because we have inhibition for 10, 20, 40, 80, and 1 to 60. And again, negative, and here we are close to the cutoff of protection, which is, I mean, a low positive samples. It appeared to be very easy, but if we check more in detail, you can see that there are some well, as this well, which is partially hemagglutinated and partially not hemagglutinated. So how we can define? I'm more than sure, in case that I have 10 technicians in my lab, 5 of those can read 20, 5 of those can read 40. And it's completely different because 20 means not protected, 40 means protected. So what I would like to say with this slide, I would like to say that the hemagglutinin inhibition assay is still now a very subjective assay. I mean, everybody's confident with the ELISA, the ELISA, you have a number that the ELISA machine generate. And if we know that 1.0 is the cutoff of protection, 0.9 not protected, 1.1 is protected. In case of hemagglutinin inhibition, that's not possible. It's very, very, very subjective assays. And it could be very dangerous, particularly for those samples very close to the cutoff of protection. That's a big disadvantage, one of the disadvantages of hemagglutinin inhibition. Other disadvantage. So we say that we have to use animal red blood cells. Okay, which animals? We have checked several different animal species, chicken, pigeon, turkey, male sheep, human blood as well, and several different horses linkage, Argentinian, Italian, German, French, different French linkage, etc. So the receptor 2 and 3 and 2 and 6 on the red blood cells are sometimes quite different. I mean, some animals has more 2 and 3, other animals has more 2 and 6. But even in the same animal species, sometimes we can see that we have different amount, different percentage of different receptors. So what does it mean? It means that the erythrocytes, the red blood cells, has a big impact on the HI, and it should be defined very well before to start. It should be qualified more than defined. So let me show some results. It appeared to be quite difficult, but it's easy. So in the first column here, on the pink column, we have the results on neutralization. So 5, of course, is negative, and 220 is high positive in neutralization. So we have checked those sera using in hemagglutinin inhibition using turkey, chicken, mallard, guinea fowl, pheasant, pigeon, male sheep, horse Italian linkage, horse Argentina linkage, and humans erythrocytes as well. And as you can see, there are some samples very, very with a good repetition. I mean, always negative, etc. But there are some samples, as the number 27, which is low positive in neutralization, completely negative with turkey, positive with chicken, completely negative with other species, high positive with the horse, and low positive with the human erythrocytes. It is not, I mean, we have something even worse. The number 38 is high positive with horse Italian and completely negative with Argentinian horse. And you should consider that we have repeated these experiments at least 5 to 10 times with different blood draw from animals, etc. The results that I show are very, very, very robust, let me say. So this means that the qualification of red blood cells before to start is the most important topic in the validation process that we should have. But even in case of the same species, we could have some, I mean, in this case, not so high, but we could have some differences. In this case, we have analyzed for H1N1 hemagglutinin inhibition using different red blood cells from 6 different turkeys, the number 4, 6, 7, 11, etc. And you can see that the turkey's number 11 is not consistent in the results as the other ones. This means that in case of clinical trials, this turkey should be discarded because, I mean, it cannot guarantee the same performance that the other ones. So as you can see, is always with titer higher than usual. So again, that's to emphasize the role of the red blood cells, and we have to say that the red blood cells must be qualified very detailed before to start with the analysis. Let me go ahead with other disadvantage. We say that a titer 1 to 40 is usually established as a correlates of a cutoff of protection when was established in 1972. As you can see here, there's a paper published by Hobson in 1972, following some efficacy trials, human challenge modern efficacy trials, using some influenza strains that, I mean, are completely disappeared now. And they established then an HI titer between 18 to 36 was associated with the 50% of protection. Do you really believe that today this value could be still valid, or we have to evaluate? I really believe and really think that it is not yet valid, and we have to reestablish something. But it's very difficult because, I mean, as you know, the influenza strain change year by year. This means that any year we have to reestablish the HI titer using, it's impossible, of course, to do any year a human challenge to reestablish what was established in 1972. So very recently with the colleagues, with the colleague in Siena, not very recently, more now it's more than 10 years ago, with Steve Black in Siena, we published a paper on Pediatric Infectious Disease Journal where we established, we did a meta-analysis for HI titer in children. And in the end, I don't want to boring you too much, but if you want, you can read the paper, of course, where we say that the data in the paper strongly support that a cutoff of 1 to 110 measured 21 days after the second vaccine dose may be used to predict 50% of clinical protection rate in this age group, which is the children. I mean, can you realize what can happen if we consider 1 to 110 instead of 1 to 40 for a clinical trials to release an influenza vaccine for children? I mean, my feel is that probably 50% of the actual vaccines in the market could be wide world and are not valid for our vaccination strategy. So again, 1 to 40 is something that we need, the regulatory people needs, but I don't believe that 1 to 40 is always valid as a cutoff of protection. One comment that I can add, but this is my personal view, is that we should consider more than a cutoff, something as the fold increase. I mean, if a subject pre-vaccination to post-vaccination has a titer greater than 4-fold increase, this means that in this case, probably the vaccine works. I mean, I'm not interested in the titer after the vaccination because I'm not sure that the titer after the vaccination as 1 to 40 is the right titer to determine the cutoff of protection. Okay, let's go ahead with neutralization. This slide is try to summarize the neutralization that everybody who's confident with the lab procedures knows very well. I want to emphasize only 2 blocks here, this one and this one. What does it mean? So the neutralization is an easy procedure. We have had the sera with probably antibodies to the with some dilution with the MDCK cells, and adding the viruses, of course, in case that in the sera we have antibodies bind the virus that cannot infect the cells after some times of incubation at a certain temperature, etc. So after that, we have to read the neutralization. And when we have to read and how we have to read. We can read overnight with no addition of trypsin at 37 degrees using, for instance, running, for instance, an ELISA versus the nucleoprotein. That's one procedure. Otherwise, we can add the trypsin and we can read the neutralization 3-5 days after the incubation with viruses, reading the cytopathic effect and not with the ELISA. So both assays are valid, can be considered valid. In the WHO manual is recommended the short methods, of course, but also the long methods with the trypsin can be used for the vaccines evaluation. How the neutralization correlates with HI? I mean, the study of correlation between HI and neutralization becomes much before than my career started. Even in 1990, some scientists tried to correlate the neutralization titer with the HI titer. And what I usually say to my students is that when they ask me, but the correlation in the end, we have a good correlation or not? It depends. It depends for which strain we use, because for some strain, as for instance, in this case, I think that this is an H1N1 strain, we have quite good correlation. In this case, that is an H3 and 2 strain, we have a quite bad correlation. And this kind of experiment has been repeated several times. Even us, together with Maria Zambon at London HPA, we did some similar experiment for H3N2 some years ago, and for H5N1 as well. And as you can see, for the H3N2, the correlation between HI and neutralization is not bad, is 0.8, and the correlation for HI and the neutralization for pandemic H5N3 is really, really, really bad. Why it's bad? And I have to point out in this direction. Is bad because sometimes, but we will discuss in a couple of minutes, the HI does not work for the pandemic strain, as we have already said before. But we will come back to this again. And that's the correlation, again, that we have established. This paper is quite recent. We published together with the NIPSC group in 2023. You can see the results for the FluCop consortium, where you can see that the correlation between the short method neutralization and the HI for some seasonal strains are not bad. I mean, this kind of correlation with the person around 0.9, it's not clear, I'm very sorry here, but it's around 0.9 or close to 0.9, the correlation is usually quite good. The situation is completely different if we use HI and neutralization long method. Long method means with the addition of trypsin and reading with the cytopathic effect after 3-5 days. As you can see, the correlation in this case is completely different, and sometimes we don't have correlation with the HI. So I have to spend a couple of words on that, because some of you can ask why, what's happened in the long time neutralization. So in the short time, we titer only the antibodies that block the viral entry. In the long term, we permit the virus to entry in the cells, and we titer all the antibodies that bind around the viruses, even the antibodies against the stalk of hemagglutinin, for instance, or versus the neuraminidase. We titer all the functional antibodies that are able to neutralize the virus. For this reason, the concept is quite different. I mean, it depends what we want to titer. When some companies come to us and they say, which kind of neutralization we have to do? It depends. It depends what you want to titer. If you want to titer antibodies that block the virus, you have to use the WHO short methods. If you want to check all the antibodies that bind the virus, you have to check with the cytopathic effect neutralization. I mean, it's a completely different concept. 2 slides only on the SRH, because we mentioned before in the list of the assays, the singular radial hemolysis is a complement fixation assay. It has been used recently for the titration of antibodies versus H5, because there was a paper published by John Wood and other colleagues which say that the singular radial hemolysis assay was suitable to detect antibodies versus H5 and hemagglutinin, considering that sometimes the HI can fail because the erythrocytes cannot agglutinate, are not enough to agglutinate animal erythrocytes with the H5N1 influenza strains. So again, the disadvantage is the same that before. The correlates of protection as 25 millimeters square. In this case, is not 1/40, but it's 25 millimeters square of area around the well is considered an area around the 25 of hemolysis. It's considered as protected, but was established in 1984 using some strains, URTS 77, etc., that are not circulated since a long time. So again, it should be reestablished. Of course, we have used, and in Europe, the European Directorate for Quality of Medicines asked us to correlate SRH at least inter-laboratories, saying that there's not a lot of laboratories in the world that are able to do SRH. You have to teach, and we teach some laboratories. And in the end, we did an interlaboratory seasonal trials correlations. And as you can see, even the labs that was teach in a short time, in the end, they are able to do this very easy assay with a quite good correlation with the expert lab. We are the lab Y, which was considered as an expert lab, and the other labs are considered the comparator labs. And you can see here that the correlation is not bad. So let me go quickly in this important part, because that's probably one of the most important things. What I already said is that neutralization is good to titer functional antibodies, even for pandemic strains. But for pandemic strains, now let me go in the procedures here. Here we have to use live virus. Just to use live virus, this means that we have to use high containment lab, and there's not a lot of high containment labs. So what we did in 2010, together with Nigel Templeton at those time UCL in London, we set up some pseudotype. What is the pseudotype? It's a false virus with a spike of hemagglutinin, with a scaffold of Gag and Paul from lentiviral, and with the luciferase as a reporter. I mean, this kind of false virus can infect the cells because it has the hemagglutinin that can attach the sialic acid on the surface of the cells, but there's no materials, genomic materials inside. I mean, it can stop the infection after one cycle of infection. And you can use this virus as a live virus to do the neutralization. And it's very easy to read. Even some students can read some luminescent cells under the microscope. We used for an official clinical trials as well versus H5N1 influenza vaccines. I reported here the number of clinical trials, and we compare the pseudotype neutralization with the classical neutralization we have biosafety level 3 lab, and we did using live virus too, and with the SRH and with the HI titer. So as you can see, the correlation is sometimes good with the classical neutralization, sometimes a little bit not so good. But in the end, we try with our biostatisticians to establish a cutoff of protection as well, which was around 1 to 357. I mean, the pseudotypes becomes quite popular now with the COVID. There are two different procedures to produce pseudotype. You can produce a pseudotype using lentiviral Gag and Paul, or using, for instance, VSV, or now very recently we have several other platforms to use to create the scaffold around the spike that you want to use. So that's a very important topic, because with a pseudotype, you can generate a pseudotype. You don't need the virus, you need only a sequence, and with a sequence you can have a plasmid. With a plasmid, you have to transfect your 293 T cells, and then you can create a false virus with a spike that you want. If you want to check your sera samples versus a virus circulated in 1919, for instance, it's almost impossible to find the live virus 100 years old, but you can generate it. You can generate a false virus, a pseudotype. So it's a very, very good opportunities for serology in the future. We are plenty of papers published comparing the pseudotype with other classical assays. I mean, that's a publication specific for the B strains, because sometimes the B strains cannot work very well with SRH and with AHI. And as you can see here, the correlation with B Yamagata linkage is a little bit better than for the B Victoria if compared with the single radial hemolysis, and the same results we got using B Yamagata and B Victoria pseudotype neutralization in comparison with the HI. So, I mean, the pseudotype, if you Google pseudotype, if you go on PubMed and you check on pseudotype, you can find a lot of papers now. But I'm pretty sure that in the future, the pseudotype, I mean, becomes very, very important on the serology, are already important in the serology assessment for vaccines. So last part is on the neuraminidase. usually say the neuraminidase, the forgotten antigen, because it's not very frequent to speak about the neuraminidase when we discuss about influenza. But it's not, from my point of view, it's a mistake, because the antigen epitopes of influenza neuraminidase have been very, very good mapped recently. So we know almost everything about the neuraminidase. When I started with my career in 1999, it was not easy to find the neuraminidase papers published. Now we are plenty, and we know almost everything about the neuraminidase, the role of neuraminidase, etc. If we want to titer antibodies that bind the neuraminidase, should be easy, but in fact, it's quite easy. I mean, we need only an ELISA. A very easy ELISA that can be set up is called ELA, but that can be set up in any labs. The concept is very easy. We have the phytoin to coat the plates. We add the neuraminidase. The neuraminidase remove the third sialic receptor. There be exposed the third galactosidose residuals. So we add the HRP conjugated lectin, which bind the third galactosidase, and in case we have the colored plates. So the concept is very easy, but we need to choose the appropriate neuraminidase, because it's not easy to have synthetic neuraminidase set up as it. So in case that we use the virus, the hemagglutinin can cross-react, and we have an action of hemagglutinin instead of neuraminidase. What can we do? Which are the solutions? The solutions are to create a reassortant virus as an H11N2, for instance. H11, we cannot expect the role of H11 because we don't have antibodies versus the H11. I mean, this procedure is quite good, but the procedure to create a mismatch virus is patented by a company, so it's not available on the market. Sometimes it's not easy. We have to request for the royalties, etc. Otherwise, there's an old method established in 2010, is a biochemical method with a Triton-X. The Triton-X usually cover the hemagglutinin, but I don't like this method a lot because it's quite obsolete. And I mean, we cannot use today this kind of biochemical methods, artificial, let me say, biochemical methods. The real solution is again the pseudotype with the neuraminidase, homologous neuraminidase, and heterologous hemagglutinin. And this is what we usually do and what we usually use to do the analysis versus to analyze the immune response versus the neuraminidase. So that's one of the papers that we have published in 2019 before the COVID, using the lentiviral pseudotype as an alternative to reassortant or Triton-X treatment to titer antibodies versus neuraminidase. And if you check only the last three columns here, you can see that neuraminidase N1 with Triton-X with pseudotype or with mismatch H7N1 virus, the results using the pseudotypes are always comparable with the other. This means that, as I have underlined here, the data support the use of a pseudotype, pseudovirus in this case, as an attractive alternative source of antigen, and justify further investigation to improve stability of these antigen sorts. So I'm going to conclude, but I have a couple of what time is? I have 5 minutes more. Okay, I have a couple of comments more. So that's the neuraminidase and the hemagglutinin. It's a nice cartoon that I usually use to explain the immune response to my students. So when they say why we don't have good correlation between HI and neutralization, for instance, I can say that because with the HI, I titer only this, and this antibody. With the neutralization, I titer only the functional antibodies. In case of the green, only the green are functional, for instance, I titer only this and this. Why we don't have correlation with the SRH? SRH is a complement fixation, and we know that IgG subclasses 2 does not fix the complement, for instance. And in case that we have a couple of antibodies that are IgG2, I can titer those antibodies in HI, but I cannot titer with the SRH. So what I want to say is that it's impossible to compare the assays that we have in our hands now, because they titer different clades of antibodies. I mean, what we should do is to have a panel of assays, to use the most appropriate assays for a specific vaccine, and then the regulatory agency, the regulatory person should comment, let me say, and should critically comment sometimes the data that we have in our hands, and in case they can request additional assays. Which kind of additional assays they can ask? For instance, data coming from for cell-mediated immunity, for the T-cell response. The T-cell response, we cannot discuss now too much with the T-cell response. We don't have enough time in case we need another 45 minutes to discuss only about the methods for the cell-mediated immunity. But I want to say 2 slides only. So one is this one, is very explicative. If you check this nice paper published on Frontiers in 2022, you can see that a comparison of 5 labs using the same panel of samples, 24 samples, collected with no specific procedures, once towing, the number of PBMCs are completely different. The blue dot lab has a lot of PBMCs after towing. The red square lab is a very poor in a number of cells after towing. This means that the data, in case that we want to produce data for the T-cell response in ELISpot, in FACS, in what you prefer, before of all, we need a good procedure for PBMC collection, even before the stimulation. And then we have the stimulation. If the variation before the stimulation is about 64%, the stimulation, if not very well established, which kind of stimulation we want to do, if with I don't know, with an epitope, with a cocktail of different proteins, etc., can be between 63 and 34%. That's again the same paper than before. I would suggest, if you are interested on cell-mediated immunity for flu, I strongly suggest to download this paper because it's very explicative on the assays that we have to use for the cell-mediated immunity. But most important is that I'm more than sure that it's not very frequently to know what we have to titer with the cell-mediated immunity. Sometimes I join a couple of times some EMA and other regulatory board, and they say, we have to ask about an exploratory endpoint for the T-cell response. Okay, which kind of T-cell response? We have to ask for what? For TNF, for interferon, for EL4, EL6, EL8, EL9, EL10, EL what? And I mean, even my department is close to the immunology department, and I usually discuss during the lunch and the coffee with the immunologist. And when I ask to the immunologist, what is the difference between IL-13 and 15 after vaccination? Sometimes they cannot say exactly what is the difference. So what I say is that the cell-mediated immunity is an important topic, but before of all, we should define exactly which kind of T-cell response we expect and we want to titer. Last slide, that's really the last slide, is the multiparametric assays. I cannot say too much on that. I mean, very recently, the new frontiers are to use Luminex and MSD, because with the Luminex and MSD, with 1 drop of sera, we can analyze a huge number of variables after stimulated with the vaccination. So we have used, of course, we have 6 different Luminex and 6 different MSD devices that is working, I mean, not 24 hours, but we are close to work for 24 hours because we have plenty of requests for those. In the end, we generate a lot of data that sometimes are not easy to understand. Again, we have tried, that's for COVID, is not for influenza. We have tried to correlate the RBD Juan ancestral and RBD Delta ELISA versus MSD, and you can see, yes, that the correlation is quite good. I mean, we are plenty of this kind of results. Again, it's very, very difficult to analyze and to say exactly what we have in our hands in the end of this analysis. Conclusion. Challenge for correlates of protection and for the assays for influenza vaccines evaluation. What we need? We need, of course, for a HI microneutralization SRH, a titer that should be associated with the protection. Probably we need at least an efficacy trial with culture or PCR confirmed cases. We need a very better, well-defined correlates of protection for pandemic and for different populations as a pediatric, etc. We need more information concerning the NA, because I was quite quickly on NA, but we don't know exactly the antibodies, the role of antibodies versus NA. Of course, we have to validate, standardize, and let me add, qualified the resources that we use in the laboratory just to reduce the variations. And we have to develop novel standardized assays for novel vaccines, because that's another part that we have missed now. But in case of a vaccine, for instance, a nasal vaccine, I mean, in case of live attenuated nasal vaccines, we cannot titer antibodies with HI or neutralization. So we need new assays at least. Otherwise, we have to go with efficacy trials. Thank you very much for your attention. I'm happy because I stay in time, which is not usual for Italian people. Thank you. Thank you very much.
Thank you, Emanuele. Greatly appreciate your insights. And really, you've outlined a lot of the challenges that's been associated with influenza and assessing vaccines or even infections for quite some time. And it's nice to see that you all are focusing on that and trying to come up with better solutions. You know, one thing I'll ask you before I open it up to general questions is, you know, you analyze the assay, the red blood cells, you say these are all variables. Do you ever look at the species for which the sera is coming from? Since a lot of people use preclinical models, or they're doing, you know, flu infects a lot of different species, whether it's experimental animals or pigs or other species, does that have any influence on how these assays are consistent?
Yeah, no, that's completely true. I have my personal opinion. I'm quite worried about the situation of HI. We have spent a lot of efforts and a lot of energies and money as well to standardize, optimize, etc., with the limitation that you said and that we know about the red blood cells, etc. In 2024, I believe that it should be really easy to do at least some synthetic red blood cells. I mean, we can do the pseudotype and we cannot produce synthetic red blood cells to easy standardize to use for HI. I mean, I'm not a biochemistry. I cannot do that. I have discussed several times with our biochemistry department at the University of Siena or in Florence or in Rome as well. And they say, yeah, it should be quite easy. I mean, we can do some synthetic red blood cells to use, but nothing happened until now. And this could really solve the situation, because now the big limitation is that an animal that you use in your lab is a different animal that I use in Siena. I mean, in case that we perform analysis, for instance, for hepatitis B, we have the same ELISA kit. You can buy from your supplier and I can buy from your supplier. And the reagent set is the same. For HI, this cannot happen because, I mean, you cannot take blood from one Turkey in the US and ship to us. When I receive blood, it's gone. So,
all right, so we have some questions. I'm going to allow people to talk and ask you the questions directly. So I'll start with Jeff Sachs. If you would like to unmute and ask your question of Dr. Montomoli, please do.
Yes, thank you very much for the opportunity to listen to your seminar and ask this question. So the question was, in a lot of the graphs you were showing, the assay values look discretized. So they have a value of 20 or 40 or 80, etc. There were certainly also examples where they were continuous. A lot of labs are using the continuous interpretation of the data, which gets away from some of the issues you showed at the beginning, where it looks like, you know, one well is, you know, a quarter or half neutralized. And then you have different operators doing different things. And there are lots of ways to do this, but they all result in a continuous value instead of discrete. Is there some disadvantage to actually using all of the data that the assay technology provides to get a continuous value?
No, I don't think so. Thanks for the question. I don't think so. I know what you say because, I mean, we have reviewed several times the continuous data, as you said. I don't think that this is a limitation. I think that it could be, I mean, a good approach, but again, is something that is out of the traditional procedures. Because, Jeff, you should consider that HI, as I said, was established more than 50 years ago. And
Yeah.
every stay with the original procedures. It's really, really complicated to upgrade, to update the
Yes.
procedure for a HI. I know that, as I said before, it's easy to set up synthetic red blood cells. And as you said, it could be easy to have a continuous number instead of 20, 40, 80. Yes, I do agree with you. Yeah.
Excellent. Thank you so much.
You're welcome.
All right, the next person is Jean Danielle. Would you like to unmute and ask your question?
Yeah, thanks a lot. So looking at your last figure on the different antibodies detected by the various analyses, and particularly in terms of IgG subclasses, I was wondering whether it could be relevant to analyze also the Fc functions such as ADCC or ADCP of these antibodies. Is it immunologically relevant for the flu vaccine, or is it too complex to perform for being relevant in clinical practice?
No, it's relevant. Thanks for the question again, Jean Daniel. It's relevant, but I mean, for the clinical part, I believe that it could be not appreciated for the vaccines evaluation. I mean, for the research purpose, when we work for preclinical or for even before then preclinical for basic research, etc., we usually analyze the ADCC, ADCP response as well. But can you imagine what can happen in case that a regulatory agency has a dossier with the HI neutralization and ADCC response as well? I mean, it's very complicated to interpret, to identify the gaps in HI. I believe that could be really, really impossible to take the advantage to have the ADCC or other sophisticated, let me know, immune response as well. But it makes sense. I mean, for research, we are in a couple of European projects where we usually use ADCC and other microchips and other. Yes, I do agree with you.
I understand that it could be quite difficult, but you may find the best rate of protection, maybe, no?
You absolutely. With the new assays, you have to find probably. So the next, not the new, but then the next frontier is to use the big data generated with this kind of assays with some software for the AI that can evaluate this kind of immune response and can give a single number, a single, I mean, answer, not a number, single answer. Yeah. Because with a big panel of results, with a big table of results, I don't think that is an advantage for the regulatory agency to approve or not approve the vaccine.
Thanks a lot.
So Ingen Berber, would you like to unmute and ask your question?
Oh, yeah. Thanks for the seminar. Actually, I'm interested with the pseudotyped virus neutralization topics, and I wonder how we can determine the cutoff value for those needs for BSL-3 or BSL-4 containment laboratory. How we can determine the cutoff or threshold for the neutralization assay for pseudotyped viruses.
We don't have, but we don't have even for the classical neutralization. I mean, for the classical neutralization, sometimes in the past for the H5, for instance, they consider 1 to 40, but it's something not correct because we don't have, as I said in my second slide, there's no cutoff of protection for neutralization. So what we said is we try to compare the pseudotype neutralization with the HI and with the SRH and we meta-analyzed the immune response 1 to 40 for H5 and 1 to 25 for SRH, sorry, and 25 millimeters square for the SRH, and in the end we found 1 to 300 something for the pseudotype. But I mean, it's something very, very, very indicative. I don't put my reputation in saying that this number is correct. Absolutely. It's a pure speculation.
All right. Yeah. All right. Thank you.
Yeah, I mean, I would second that. I know in some of our animal studies, we've had undetectable newts or undetectable HI for an H5 virus, and yet the animals seem to survive challenge with very little clinical signs. So I don't know what
Are you sure, Ted, that the animal model could be translated in humans?
That's right. That's part of the issue, right? All right, we have one last question that's typed in the chat, so I'll read it to you. The anonymous person has listed, do you think these FDA and EMA approved assays and cutoff will be applicable for flu vaccines being developed for newer platforms such as mRNA? In other words, does the platform you deliver it matter?
Yes, I think so. Why not? I mean, the mRNA vaccine and the self-amplifying RNA vaccines usually elicited the immune response. So it works, the classical assays. I'm not worried about that. I'm worried for other kind of vaccines, as the live attenuated vaccines, for instance, intranasal, or there are some vaccines that are specific for the versus the nucleoprotein, for instance, and this could be evaluated only with the efficacy trials. But for the mRNA vaccines, I think that HI and neutralization could be applicable as for the classical subunit or adjuvanted or other vaccines. Why not?
Okay. So Bob H., would you like to ask your question?
Sure. I just wanted to ask, what about vaccines, and you just alluded to this, that are focused on at least a portion of their vaccination generates mucosal immunity, and what different assays might we need to look at in order to evaluate that, along with, the HA and anti-NAI activities?
We did some analysis in the past for using for the nasal vaccine. Sorry, I was distracted. So we did some analysis in the past using a traditional IgA ELISA. So again, IgA in a nasal swab or in saliva, it's quite easy. I mean, it's a method that could be easily standardized. Of course, we don't have a correlates of protection. We can consider a titer increase pre and post vaccination. What is a little bit more difficult for my point of view, but it's something that is still under discussion, because I believe that does not make a lot of sense, is to find to titer IgA in serum. So, but I don't believe, but sometimes some companies requested us to titer IgA in sera samples as well. I strongly don't suggest, to be honest, to do that. And it's very hard to find IgA. Probably we need a sensitivity completely different out of the classical ELISA. We have to find something more sensitive. But for the ELISA IgA in a nasal swab or in saliva, it's very easy. I mean, it's something that we can do easily.
Thanks. All right, Linda Brock from Pfizer, I'm going to let you have the last question for this webinar. Feel free to unmute and ask your question.
Hello, can you hear me okay?
Yes.
Okay, great. My question was more towards the beginning of your talk. You had talked about, for the HNAI assay, the 1 to 20 and the 1 to 40 for an analyst, it's difficult to discriminate the smears and or pelleting, and then being able to, with confidence, consistently do that through a clinical trial. And so my question is more around, if you use the FLUHEMA instruments, and then if you have, in your experience, have you found this to be more reliable? And just in general, if you could comment with the use of that technology.
I don't know, if you could hear that well. I could read
No,
it
no,
to
but
you.
I read the answer on the messages. Yes. So we have tried to use some instruments, not only the flu HEMA instrument, but also other instruments that are, I mean, there's a company in Colorado that is doing some reader for HI. I mean, it works, but we still need the human presence because it's very easy that there are some fail in readings. I mean, it's different than I usually compare with the ELISA. When we have one ELISA well, it's easy to understand for the reader if it's yellow or a light. In the case of hemagglutination, sometimes we don't have a very good experience with the automatic reader for hemagglutination. But this is something where there are several companies that are working around the world. Not only the flu HEMA, there are the other one is Indivir. There's another company which is called Indivir, which has an automated reader for the HI. It works, but we need the human presence. There's not an advantage. In this case, we can read using our eyes, to be honest.
Okay, thank you.
You're welcome.
All right. Well, thank you, Emanuele, for your presentation. I appreciate the lively conversation and questions. This will be posted on the ISV website as a YouTube video for those who may have missed it or would like to watch it again. We'll look forward to seeing you next month. Dr. Gunvig Groland from the University of Oslo will be presenting on developing vaccines for the next pandemic. Until then, enjoy and be safe. We'll see you next month. Thank you.
Thank you. Bye-bye. Thank you. Bye-bye. Thanks.
Thank you.